Hello, everyone. My name is Laura. I'm a PhD candidate in Julius Lux's lab at Northwestern University, and I'm really excited to be sharing my research with you all today. Especially thank you to Jill for inviting me. And yeah, I'll be talking about the effect of sequence variation along along pseudoknot base pairing within the fluoride riboswitch. So, okay, maybe the laser is a bad idea. Okay, so I just wanted to briefly go over some cell biology for those Eterna players who maybe just don't have as much familiarity. When I think about RNA and RNA structure, I really think of it in this overarching content of what the cell is doing. So I like to think of the cell as a recipe card where you have your DNA as a cookbook, and then your RNA is a recipe card that you can copy down from the DNA, the cookbook. And this recipe card is what you can carry around the kitchen or even to the grocery store to grab your ingredients. And then once you have your RNA or all your ingredients from the grocery store, you can make proteins in the cell or a cake in the kitchen. Personally, Black Forest cakes have been on my mind. And just to contextualize this in society a little bit, this is how the mRNA vaccines came together, where Moderna basically created this recipe card describing just a portion of the SARS-CoV-2 virus that our immune system could recognize as not part of our bodies and learn to attack it and recognize a real infection and be better prepared for that in the future. So when getting ready for this talk, I wanted to play around with the Eterna myself because it's not something I've had much exposure to. So I added my favorite sequence, the fluoride riboswitch, and saw this beautiful structure that was a lot of fun to just piece together and give the dot bracket notation. However, a key feature that was missing was the pseudoknot, as has been coming up in several of these talks. So that was something that I added in myself. Or when I think of the fluoride riboswitch, I put it together in this 2D schematic where I actually centralize the pseudoknot in the middle of it. And when I look at the structure in three-dimensional space, this is how it kind of all comes together with the pseudoknot now in this rainbow nucleotide color formation from front to back, just all put together there. Now, what I really like about this 3D structure is how it also reminds me how in 2D representation, it's really hard to get some of these nucleotide interactions. So I denoted some long range interactions with these light lines around the 2D representation. But in the 3D representation, it's really clear to see how close together they are to form such stable base pairing interactions. So I always like that reminder. Keep my eye on the chat. Yes. Oh, great. Thanks, Jill. So the fluoride riboswitch, speaking about multi-states, the fluoride riboswitch roughly exists in two states. Here I have the 3D structure up top, just so we can remind ourselves what the pseudoknot looks like in real life, more or less. And the 2D easy to view schematic here, where the red nucleotides in 3D are oriented this way, but in my 2D schematic, I have them up top here. So when the fluoride riboswitch is being made during the process of transcription, I don't have my DNA here. Please forgive my overly simplistic diagram. As the polymerase is transcribing, in the absence of fluoride, a structural rearrangement is going to occur that causes this termination hairpin to form. The polymerase leaves the DNA, and ultimately you stop making the gene that would ultimately lead to a protein. However, in the presence of fluoride, the pseudoknot and overall structure are stabilized against the structural rearrangement occurrence. The polymerase keeps on transcribing as if nothing happens, and you're able to get your expression of protein. Now, the two-state feature of RNA resides in what we think of with the equilibrium folding and thermodynamics of RNA sequence and structure. We know that if we have a single RNA sequence, if we had 100 copies of it, not all 100 molecules would form the same structure. Rather, they would form this ensemble pool of different structures over some favorability towards one structure or the other with these various kinetic parameters to overcome to get into different structural states. But that's thermodynamics. I'm really interested in the kinetics of RNA folding and how that influences the structural switching process. So when I think about RNA folding, I think about how the secondary, these local interactions, these hairpins can form rapidly at the micro to millisecond time frame, whereas these tertiary interactions like pseudoknots or long-range interactions can take seconds to form. Notably, these long-range interactions like pseudoknots are happening at the time scale of transcription, which is also at this like seconds time frame, which means that as transcription is occurring, RNA structure is already starting to form. However, transcription is not a unilateral process. In fact, there are these encoded pause sites throughout DNA and genes that let the RNA, the polymerase pause, slow down, and these pauses can last again on the seconds time frame. During these pauses, the RNA can take advantage of that and actually undergo structural rearrangement to form whatever new or more favorable structures there are. So really, when I'm thinking about the fluoride riboswitch, I'm thinking about this out of equilibrium transcriptional folding two-state mechanism that's going on. And this is what my lab gets really excited about in studying is how does a simple ligand, as simple as the fluoride ion, influence this juncture point between when the fluoride riboswitch is either going to undergo a structural rearrangement and stop transcription, stop this whole cellular process, or allow the stabilization to be influenced by a single ion and allow the cell to keep making this gene. We hypothesized that, frankly, it was due to the pseudoknot, as in the title of my talk. So I've updated the color scheme a little bit here because the process I'm going to talk about, it's really going to be centered on this central strand of RNA that either participates in the pseudoknot or participates in the new terminator hairpin that forms. And basically what's happening is I've now used the color coding to denote like similar nucleotides. So the idea down here would be these purple nucleotides could both be Gs, both competing for the same C. So while the C was originally in the pseudoknot, if it's successfully displaced or stolen away by the new invading strand, then that's how we can get this structural rearrangement. Now, if this hypothesis is true about the fluoride riboswitch and pseudoknots, can we identify the sequencing rules governing the switching process so that we can design better or even novel functional RNA structures in riboswitches? So to investigate this sequence variation questions, I developed a cell assay where we just delivered DNA that had instructions for making a fluorescent protein that we could measure and deliver it to the cells and have the cells express this fluorescent protein, which we could then measure using flow cytometry. So here I have a control that doesn't have a riboswitch. So this way I could determine how fluoride was impacting my cell systems. And I see that there is about a 1.1 fold change in the presence of fluoride, this sodium fluoride condition. And that just lets me know that, okay, sometimes fluoride does have some effect on my cell system, but it's pretty negligible to what I would expect of the fluoride riboswitch, which I am showing here. That's supposed to be fluoride riboswitch shows fluoride can regulate. So sorry, my cell is covering up the word show. But I had this standard fluoride riboswitch sequence from Bacillus. And when I put it in my cell assay, I see that one, I'm overall getting a lot less protein being made, but two, in the presence of fluoride, now I'm getting that 2.4 fold change of fluorescence in the presence of fluoride, letting me know that the riboswitch is in fact regulating the amount of fluorescence being produced. But I called that the standard version of the fluoride riboswitch, specifically from Bacillus. And the reason why I had to specifically name the sequence was because there are actually thousands of versions, well, 1,600 versions of the fluoride riboswitch. When you look across evolution, these have been computationally identified and not necessarily high throughput studied in the lab yet. Here I'm showing a phylogenetic tree. At the center is the root, and then there's just so many species that they just put the whole tree into a circle. Over here is where we have Bacillus, the standard version I was just talking about. And on this other side of the tree, we have Thermotocopetrophylla's version of its fluoride riboswitch, which was the version that was used to solve that crystal structure I was showing earlier. I've highlighted here in orange just some various nucleotide differences between the TPE version compared to the BCE version. This species is one that optimally grows at 80 degrees Celsius. So when thinking about RNA sequence variation, I really like to bring back in the baking analogy because I know when I'm baking, if I slightly alter how much flour, not fluoride, flour or sugar or the types of sugar, baking powder, I can actually get slightly different variations of my cookies that are going to taste or even look different. But why would we expect that of our cells? Or similarly, like The fluoride riboswitch looks the same, whether it's between BCE or TPE. So we would realistically expect that they should function the same if sequence equals structure equals function. However, as I'm sure you could tell by that lead up, the TPE fluoride riboswitch didn't really work in our system. We see that we're getting a ton of protein being made and really no big difference of fold change in the presence of fluoride, letting us know that the TPE fluoride riboswitch at 37 degrees Celsius isn't really regulating gene expression at all. We hypothesized that this was just simply due to the TPE riboswitch being evolved to optimally function at 8 degrees Celsius, and it just needed more energy to undergo the structural rearrangement process. So to again center ourselves in the central hypothesis of this project was the fact that there was a structural rearrangement going on through the pseudoknot that was impacting the ability for the riboswitch to turn on or stay on. So in order to use these two fluoride riboswitch variants to test this hypothesis, we simply took the pseudoknot in its overlapping sequence and just switched them between the two riboswitches to make these chimeric variants. So when we have the TPE fluoride riboswitch with the pseudoknot from Bacillus, we see we're getting a much less protein expression being done, and we're able to regulate the amount of gene product being made in the presence of fluoride. When we do the same with the Bacillus fluoride riboswitch, but now with the pseudoknot from TPE, we see that we're getting a ton of protein being made. Please notice the different y-axis, as well as the fold changes now mimicking the fold change from the original TPE fluoride riboswitch. This really solidified in our minds that it was the pseudoknot and its sequence that was really impacting the ability of the fluoride riboswitch to function. Yes, thank you both. So for the first half of my PhD, this was really exciting to start looking into. And of the pseudoknot, there are 6 base pairs and overall 18 nucleotides that could be contributing to this phenotype. And what all my research distilled down to was how it's just a single base pair. And that single base pair is up here on the top. So in Bacillus, the top of the pseudoknot is a GU wobble base pair. Riley briefly got to mention them, gave a shout out. I was like, oh yay, I love my wobble base pairs. In Bacillus, we changed them to a canonical GC interaction, where we then noticed that the amount of protein was the same regardless of fluoride. So Bacillus lost the ability to regulate the amount of protein expression just by that simple single nucleotide change. In TPE, we were able to do the opposite, where it went from a canonical GC base pair to then converting that into a wobble base pair. And what we found was we overall lowered the amount of protein being made, suggesting that in both conditions, more structural rearrangements were occurring. And in the presence of fluoride, we're now getting that full differentiation and letting us know that the riboswitch is better able to respond to the presence of fluoride. However, I mentioned that the TPE fluoride riboswitch was optimized to evolve to optimally function at 8 degrees Celsius. So I wanted to develop an assay that would allow me to basically test that. And now, unfortunately, I couldn't just take my E. coli cells and heat them up to 8 degrees Celsius. They wouldn't like me very much or work very well. So what I did was I took the transcription system out of the cell and just artificially recapitulated it in a test tube, where I took a RNA polymerase from another thermophile, Thermus aquaticus, which I could then heat up to 65 degrees Celsius to do transcription. So almost doubling the transcription temperature that I was allowing it to happen in my cells. And after running transcription in a test tube, I could take the RNA, run it on a gel where I could look at the band length. If it was a short band, I could interpret how a termination event occurred, versus if it was a longer length strand of RNA, then I would interpret it as no rearrangement occurred and we got the full length product. Transcription continued on as per usual. With the wild type TPE sequence, we see that in fact, we do get a lot of that read-through product with a little bit of that termination product without the presence of fluoride. And we get less and less terminated product over time. When we have that single point mutation, we see a very similar trend, but now we're getting a ton more termination product in the absence of fluoride that slowly disappears as we add more fluoride, showing that even at these elevated temperatures, this local nucleotide interaction of this wobble base pair is far more important than the amount of energy that exists within the transcription system. Now, It was pretty hard to believe that out of the 18 nucleotide space, it really came down to this one base pair interaction. So to investigate that further, I went back to my E. coli system so I could do counter experiments in the cells. But sticking with the transcription assay, I'm now doing transcription at 37 degrees Celsius. What that looks like for the TPE wild type is roughly 50-50 termination, anti-termination. But again, no recognition of fluoride really determining how much with the length of the RNA. So I made 3 series of mutants that either maintained a wobble base pair and just went down the pseudoknot. So in the second position of the pseudoknot NTP, it's a canonical GC, which would have 3 hydrogen bonds. We changed that to be an AU base pair interaction because that would be 2 hydrogen bonds. We hypothesized that we were weakening that position of the pseudoknot, weakening that position of the structural rearrangement, and we should be able to better understand how each position along the pseudoknot was contributing to the structural rearrangement. We see with the wobble base pair, even with that weakened interaction, we're still getting pretty good switching recognition within the transcription system. However, without that wobble base pair, then that rearrangement is just not occurring, and we're back to 50-50. We're back to indiscriminately whether or not fluoride is there. The transcription is happening the same way. In the next variation we had, we went down another peg of the pseudoknot, and we see the same phenotype between the wobble base pair and the no wobble base pair variants. And the same is true for that last mutant. So overall, this really highlighted how the structural switching ability of the Fluoride riboswitch really came down to a single nucleotide in a particular location. This was really interesting to us, and we were wondering why this nucleotide, why this position. So we collaborated with Professor Alan Chen at the University of Albany and his student Elise White, who do all atom molecular dynamic simulations of this switching process. So I have arrows here orienting ourselves. So the wobble base pair, this is the BCE wild type sequence. So the wobble base pair is right here. And in the movie I'm about to show you, the position is right up here in the top. What you're going to notice is the uracil is just going to almost immediately rotate out of the pseudoknot. That's going to have a downstream impact of relaxing the overall structure, allowing this new invading strand to do what I want to do, which is get into that low free energy state and base pair with the other strand. So here we see the uracil just completely took itself out of the question, out of the equation, and that allowed the new nucleotides to come in and form with the previously formed structure. And it's really a rapid process. It's 6 base pairs. It doesn't have a lot of things to move around. So it happens and it's done, which works for the time scale of transcription that this riboswitch is working on. However, now we were able to do a model for the version of the fluoride riboswitch with BCE that is canonically a GC interaction. And what you're going to notice is that C is really locked in tight with its G, and it's not going to rotate out of the pseudoknot. The whole structure isn't going to relax, and the new nucleotides are just going to be kind of stuck as the odd man out. So we see here the C and G really tight together, and then the new nucleotides don't really have anywhere to go. So this kind of lets us know how the pseudoknot could be so stable against a rearrangement, especially on the timescale of transcription, to allow the process to continue, the transcription process continue on without the structural rearrangement occurring. So in this way, I really think of this wobble base pair almost as a reclining chair where the wobble allows there to be kind of a lever on the pseudoknot structure, allowing it to dynamically engage and go back and forth. Whereas if it's a canonical GC, it gets stuck and it's really difficult to rearrange and move around. Now, I don't want you to just think that this is just fluoride, and I was really interested when working on this project, learning about other instances of the wobble base pair having such a structural rearrangement impact on RNA structure. Now, I'm showing some work done by my lab a few years prior where we were studying the SRP RNA molecule. And what Angela observed was within the co-transcriptional folding of the SRP molecule, between nucleotides 109 and 110, this massive structural rearrangement process underwent, where you went from 3 distinct helices to 1 overarching long helix. Notably, there was a wobble base pair within that first helical stem that allowed dynamic helical formation. But when they mutated the U to a C and locked that hairpin nice and tight, all of a sudden there was no longer this rearrangement that occurred between 109 and 110 nucleotides. It was kind of locked in this pre-formed structure. And I know Eterna is getting pretty interested in riboswitches, and I also wanted to just highlight the biotechnology, the societal impact of this research, especially coming out from my lab where Walter was able to actually take the fluoride riboswitch, and this is how we came with BCE as being our standard. He was able to take this and enact a field deployable water sensor. So most of the time, some fluoride in our water is good. It's great for our teeth. It strengthens it. But if it's above a certain EPA limit, it can actually lead to damage. Now, we normally don't have to worry about that, but for people who are getting their water from natural resources, fluoride contamination is an issue that they do have to think about. And so Walter was able to take the fluoride riboswitch and make this very cheap, easy to use sensor that we were actually able to test out in Costa Rica and show that it worked to detect fluoride levels above the EPA recommended limit. So just some really exciting work going on with water contamination and synthetic biology and the space that there is in this work to bring our products to, or bring our science to helping people. With that, I would like to thank everyone in my lab for participating with this project, giving me feedback even on this talk, and just being great overall people to work with, my collaborators who have been so generous with their time and their help with this, and just overall to Julius for leading me through this project. With that, I can take any questions, and I do have one final RNA rearrangement movie that I figured people could watch as questions were coming in. But thank you for your time.